And dear students of 12th class, today in your botany we are going to discuss the general DNA for you, and it is historic. What were the historical progresses that were made in the ultimate discovery of the structure, the final structure of the DNA? What were the historical contributions of the different scientists or the geneticists who by whose approach it was clear that how the DNA should look like, should be like, and ultimately its structure was unveiled by the two scientists, which we are going to have a look upon. This is the historical approach or historical contributions made by the different geneticists. And firstly, a general introduction of DNA. This DNA is a long polymer of deoxyribonucleotides. In fact, DNA stands for deoxyribonucleic acid. DNA is a long polymer of deoxyribonucleotides. The length of the DNA is usually defined as number of nucleotides or a pair of nucleotides referred to as base pairs present in it. This also is a characteristic of an organism, that is the number of base pairs. It is fixed for a particular organism or a particular type of species. And then, for example, a bacteriophage known as Phi 174 has 5,386 nucleotides. Bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 to the power 6 base pairs, and the haploid content of the human DNA is 3.3 into 10 to the power 9 base pairs. And let us now see what were the historical contributions made in the, what were the progresses made in the ultimate discovery and the structure of the DNA by the different scientists. Nucleic acids were first isolated by Friedrich Miescher in 1869 from pus cells, and they were named nuclein. And then Hertwig in 1884 proposed nuclein to be the carrier of nucleic acids, or rather, Hertwig in 1884 proposed nuclein to be the carrier of the hereditary traits, and because of their acidic nature, they were named nucleinic acids, and then nucleic acids by Altman in 1899. Fischer in 1880s discovered the presence of purines and pyrimidine bases in nucleic acids, and then Levine in 1910 found deoxyribose nucleic acid to contain phosphoric acid as well as deoxyribose sugar, and these were the contributions. And then he characterized 4 types of nucleotides present in the DNA. In 1950, Shergaff found that purine and pyrimidine content of the DNA was equal. By this time, W.T. Astbury had found through X-ray diffraction that DNA is a polynucleotide with the nucleotides arranged perpendicular to the long axis of the molecule and separated from one another by a distance of 0.3 nanometer. In 1953, Wilkins and Franklin got very fine X-ray photographs of DNA. The photograph showed that DNA was a helix with a width of 2 nanometer. And one turn of the helix was 3.4 nanometer with 10 layers of the bases staked in it. And then Watson and Crick, 1953, worked out the first correct double helix model from the X-ray photographs of Wilkins and Franklin. Wilkins, Watson, and Crick were awarded the Nobel Prize for the same in 1962. And Watson and Crick, 1953, built a three-dimensional molecular model of DNA and that satisfied all the details obtained from X-ray photographs. They proposed that DNA consisted of a double helix with two chains having sugar phosphates on the outside and a nitrogen bases on the inner side. The nitrogen bases of the two chains formed complementary pairs with purines of one and the pyrimidine of the other held together by hydrogen bonds. In fact, the purines and pyrimidines were linked as adenine with thymine and cytosine with guanine. That is, to remember it simply, it is AT and CG. A complementary base pairing between the two polynucleotide chains is considered to be the hallmark of their proposition. It is, of course, based on early finding of Chergaev that adenine, thymine, cytosine, guanine. The second big proposal was that the two chains are anti-parallel with 5' to 3' orientation of one and 3' to 5' orientation of the other. The two chains are twisted helically just as a rope ladder with rigid steps twisted into a spiral. A rope ladder with rigid steps twisted into a spiral is just like the DNA molecule looks like. Each turn of the spiral contains 10 nucleotides. This double helix or duplex model of DNA with anti-parallel polynucleotide chains having complementary bases has an implicit mechanism of its replication and copying. Here, both the polynucleotide chains function as templates, forming two double helices, each with one parent chain and one new but complementary strand. And this phenomenon is called the semi-conservative replication. In vitro synthesis of the DNA has been carried out by Kornberg in 1959. And to know about the structure and the other these complications regarding the structure of the DNA, we will try to see the open structure of the DNA, how the different what lies behind the linkages of the different molecules that are held together in the double-stranded DNA helix. Thanks. Have a nice day.